everyone. Welcome today to the next instalment of the MPCCC and Monash BDI Cancer Discovery Seminar Series. I'm Mark Shackleton, and alongside Professor Melissa Southey, I'm a co-director of the MPCCC. So I'd like to open today's seminar by acknowledging the traditional owners of the lands on which we're joining together from today, and respectfully acknowledge their elders past, present and emerging, as well as any members of the Aboriginal and Torres Strait Islander community gathered amongst us here this afternoon. So the Discovery Seminar Series is a community of scientific researchers, health professionals and academics from all across Victoria who work within the cancer research and clinical care sectors. And the seminars were created originally with the idea of helping us to share advances in the latest to share the latest advances in research as well as clinical care across the MPCCC partnership, but also more widely. And of course, we've had speakers from really all across the state, indeed country. In fact, we've even had international speakers, of course, and across a community that's really interested in communicating new ideas and learning about new approaches, current as well as potential future ones, to caring for cancer patients. Today's seminar is going to cover the role of molecular pathology, which is incredibly and increasingly important in the management of cancer patients, and certainly is really helping to redefine the pathological diagnosis of many different cancers, but I think particularly primary brain tumours, and many of the developments in this field have really changed the way we think about this group of diseases substantially. And we're extremely lucky today to have one of the international leaders in this field, Professor Catriona McLean. Catriona is currently Professor and Head of Department of Anatomical Pathology at the Alfred Hospital, a position that she's held for nearly 20 years. In this capacity, she also heads the Solid Cancer Molecular Pathology program. And in her spare time, she's also really got an international reputation as an expert in neuropathology. She's a director of the Victorian Neuromuscular Service and also director of the Victorian Brain Bank, and a diagnostic pathologist for the Florey Institute. Additional to that, she's also an honorary neuropathologist for the Royal Children's Hospital, the National Creutzfeldt-Jakob Disease Registry, and also the Australian Phenomics Network. So a very, very busy person, and I can vouch for that. She's incredibly good to work with, but always under pressure because of the number of people that she has hassling her for her expertise. We've got it for the next hour, which we're very grateful for, Catriona. Thank you very much, and I'll hand over to you.
Okay, great. Thanks, Mark. Well, I thought it was quite fun to put this talk together, and I was looking forward to doing it because, you know, there's been a lot of changes, as Mark says, in the realm of neuropathology and molecular techniques. But I just thought, because often in some of these presentations, we've got quite a wide variety of people. So I really wanted to focus and make sure people understand what I do as a pathologist, because sometimes I get the impression from some people that they're not quite sure what a pathologist does. So from my point of view, the main aim of pathology is patient care. That's what I'm here about. Now, people might not understand that when you look at my microscope and I'm nowhere near a patient, that's not what drives me, but it is. And patient care is number one. I want to provide an accurate diagnosis. I want to provide a diagnosis that is in keeping with world standards. I want to provide a timely diagnosis. I want to provide prognostic information. And I want to interpret as much pathology data that I can to provide the most full and report that I can to the clinicians who are actually going to provide the care in terms of management. So I just want to bear in mind that we're still here and patient care is important. So for the purpose of the talk, I asked myself, what can the molecular pathology provide in the space of neuropathology? And what is becoming, it is an exciting field. It's very challenging, and you have to continually update your information. But it adds additional diagnostic information to what I've got from just looking down the microscope. So it is improving my accuracy of diagnosis. That's what I think. in addition, not only does it improve my diagnosis, it also can give prognostic information. So is it worse or going to behave worse or better or whatever? And we've always provided prognostic information down the microscope. I mean, we've decided whether something's well-differentiated tumour or poorly differentiated. That's prognostic information, but this is additional prognostic information. And finally, there's that role of molecular pathology in targetable treatment options. So does the molecular techniques that you provide give us a treatment arm? Now, this is possibly at the moment, and I think it's not unrealistic to say that this whole area of targetable treatments for brain CNS kind of tumours is possibly not as forward as some of the other areas in the lung or colon or melanoma space, but it's definitely something that will evolve over time. So what we've got is a movable feast, and we're getting more and more information as we go along each year. So just to clarify with all of those who are out there who might not realise, what happens in the lab is we get brain tissue, it comes in as fresh tissue, we examine what we've been given down the macroscopically to see what, and we record that. We fix the tissue, we process the tissue, we then get an H&E stain slide, and that's what goes to my desk. And after I've looked at the H&E stain slide, I order additional tests, and on the basis of all that information, I can write my report. Another important issue, as I mentioned in the beginning, was the turnaround time. Effectively, what you can issue a report, but if you issue a report 3 months down the track, you know, it's not relevant. What you want is a report that comes out in time, which means that any additional test you do, you have to understand what the turnaround time of that test will be and how that's going to interface with your clinicians that you're dealing with. So, for instance, an H&E will take 12 to 24 hours, so that's quick. If you do additional immunoperoxidase, and that's almost inevitable in CNS tumours, that whole process will take 6 to 8 hours. So if you order it at 4 o'clock in the afternoon, clearly it's going to be an overnight run and won't be ready to the morning. So there are, it's effectively an additional day if you want immunoperoxidase. And if you want a second round of immunoperoxidase, it's an additional day. So that's the kind of turnaround time. Now, we'll be seeing that in addition to immunoperoxidase, some of the molecular testing that we're doing to improve our diagnosis is the in situ hybridization. So we've got to have the scientific staff, the machine's got to be working, the probes have got to be in, but we like to think that we can do these in 1 to 2 day turnaround time. And as you can see, for any tests that we do that we get into the department that's new, we have to have it as a NATA accredited test, which means we've got to do a whole lot of rigmarole to make sure it meets the standards acceptable for a diagnostic test. It's kind of a different framework to research, but sometimes I think that if we adopted some of these very strict protocols in the research frame, we'll probably be improving our research. So here we have EGFR, which we've been running only since September 22 to look for amplification. I'm showing you pictures of this, and it's relevant for some tumours. I'll explain why. CDKN2A-ish we're doing since September 22. And earlier, when we first started, we started doing in situ hybridization for the chromosomes 1p and 19q, and we started that in 2020. So as you can see, it's only 2020, we're 2023 now. This is every time there's more things happening, we add. You can see that there's been 25 tests here, only 8 CDNK2As, and quite a lot of this test 1p19qs. So there's the kind of framework for the in situ hybridization, and then, which is what we were heading a little bit more for, is the next generation sequencing. So currently we do 1 run every week, and so the turnaround time for that's going to be roughly 7 days. So that's the kind of perfect turnaround time, and I like to think we stick towards it, and I think we generally do.
Basically their own day. That was
so
yesterday.
There's someone who's not going to mute on. Sorry. So just the next
Sorry, Catriona, I think it's Tony Taganis. Tony, could you just get yourself off mute, please?
Hey, Tony, is he listening?
On to mute.
Okay, thanks. So the really important next step that everyone out there needs to know, and I can't stress how important this is. We do a lot of these things routinely, and the older I get, the more important I realise these things are. And this important step is every week we have a neuro-oncology forum where we discuss the radiology, the pathology, the clinicians discuss the management. And this is where things happen where if we don't have a match between the pathology and the radiology, or the clinical and the pathology, this is where we have a really good meeting of the minds. And at some stage, some circumstances, we can look and say, well, actually, I don't know that I've done the right tests, or the or my diagnosis might be wrong. And this is where we have a really important meeting. So this Neuro-Oncology Forum Monday mornings is super critical in getting us to reach the right diagnosis, but more critically from then on to manage the patient according to the right diagnosis. So this Neuro-Oncology Forum, terribly important. So what do we do in terms of our next generation sequence here? So we've got an Illumina platform. What is terribly important is that simple step of tumor selection. So when the biopsies come to us, we have to make sure that the tumor that we're selecting is got a certain percentage of tumor, otherwise we'll be getting false testing, and we don't want it to be heavily pigmented and heavily necrotic, and we want to select out relevant tumor to get the best quality high-grade tumor. And I think a lot of you who work in the molecular space will understand exactly what I mean. Currently, what we do here is a targeted panel with a whole lot of known mutations. So it's a custom designed targeted panel. It's not as comprehensive as some of the other broader panels done up in other places, but it meets a lot of our objectives. It still needs to be updated, but once again, every time we update, it's quite a big palaver in terms of making sure that we got the NATA accreditation for every update we have. So we do need to update, and Mark's well aware of that as well. But currently we do a custom design panel. It's got about 70 known genes on it with all their mutations. So it meets most of our requirements, but always needs to update. So that's just the basic framework of where I'm coming from in terms of diagnosis. So what happens next? So in the realm of CNS tumours, in the World Health Organization in 2021, 2 years ago, brought out a new manual effectively of how I, as a neuropathologist and neuropathologists all around the world, should be now integrating into their diagnosis additional tests. So this manual that we get effectively, the World Health Organization categorization of all central nervous system tumours, I think it's the 5th edition. Now, unfortunately, I'm old enough to, I think I go back to the 2nd or 3rd, but every time it comes out, it's a massive new learning, because what they do is they add in all the literature that's come in between and plonk this into the new CNS tumour categories. So what happened in 2021 is that the kind of way we reported started to change. And for me, it's very important. And what we started doing is what is called an integrated diagnosis. So in the old days, it was just the H&E, then it became immunoperoxidase, which helped us to refine our diagnoses. Now what we're doing is added in immunoperoxidase, in situ hybridization, and molecular studies. And within that framework of integrated diagnoses were some essential information. That's what the WHO said. You have to have this essential information to reach these diagnoses. And then there was desirable information, which meant you didn't have to have it, because, you know, you may not have the tests. So in this WHO CNS categorization of 2021, in addition to this kind of new way of looking and diagnosing central nervous system tumours, 22 newly recognised tumour types have been found. So in the 5 years or whatever between the two different iterations, there are 22 newly recognised tumour types. So it means that for all those 22 new tumour types, I've got to go and get accustomed to them, because they're probably very rare. Because there may have been 10, 15. I think one of them only had that kind of number in the literature. So you're not going to come across them in your daily routine practice. So you've got to get up to speed on that. So it's a big ask. It's complex stuff, and it is always changing, which makes life fun but also difficult. So let me look at the report structure so you know where I'm heading. What's important in the central nervous system is quite a few things, and one of them is the tumour site. Unlike the systemic system of like the body, like the breast or the colon, in the brain, it's not just where in the brain, it's where in the brain. So there are tumour sites in the brain where tumours occur much, much more frequently than others. So the tumour site specific is important in a central nervous system tumour. We then provide the integrated diagnosis, which has both the histology and the molecular testing. And we give it a classification according to what we think we see down the microscope. And then we also, based on what we're seeing down the microscope, we give it a WHO grade. Now, this grading system is very important to the clinicians because it tells them, based on all the criteria we know and on multiple studies that have been done over many, many years, how these tumours will probably behave. So a grade 1 WHO grade tumour is a low-grade tumour. If you can completely remove it, that is a problem in the central nervous system. It's not like other parts of the body where you can often get full removal. It will never recur, as opposed to a grade 4 tumour, which means the tumour is a very aggressive tumour and will probably lead to death in a rapid time frame. So we provide the grading system, and then we provide molecular information as well. So it's a busy report. Now, I think it's important to come to these kind of comments before we go into a bit more detail. But, you know, I've been on this committee, which is an international committee that looks at updating the central nervous system in between these new renditions of the central nervous system tumour WHO gradings. And, you know, what it looks for is interim changes that are occurring in the literature. And if some of those are really important, we want to rush them forward and say, look, you know, we need to have this come forward quickly, because we're not going to wait 5 years to tell the clinicians we've got a new test that's really relevant. So these systems are never perfect, because there's always more information that we find with time. And they really just reflect the quality and understanding of the experts in the area at the time, or the quality of the molecular tests available to those clinicians, pathologists. So no system is perfect. Every fit system is evolving. They did decide to establish this C Impact Now group so that it allowed interim changes to be made. Another important point is the molecular testing is an additional cost to any pathology department. Not every pathology department can afford these tests. And that's specifically, if we go down to my last point here, that many of these additional tests are really not available around the world. And there's been a big push because I'm also vice president of the International Society, Asian Oceania Society of Neuropathology. And they did a recent survey of all these additional tests that have been promulgated by the WHO. And places like Indonesia do not even have immunoperoxidase. So whilst the international WHO is saying this is what you have to do, in reality, across Asia, Africa, and Oceania, they cannot afford these testing structures. So we're involved in another study here in trying to help these communities get towards the best central nervous system diagnosis they can. Okay, so another point I'm making is that various tests take different times, and on occasion, the clinician really wants a fast result and they can't afford to wait for the longer test results. So there's kind of an interim kind of diagnosis has to be made. And some tests can be acting as screening tests that we can do just to avoid these wait times. So just general comments about what we do. So let's now start looking at a bit more about the tumour types. Now, this is where I hope that it doesn't get too big wordy, complex, because people don't use these words all the time, and I hope I don't lose you. I've tried to actually just keep it to a very limited group, because clearly there are many, many, many different brain tumours, and there's no point discussing all the rare ones. What I'm bringing up is the common ones in the adult world, and the adult type diffuse glioma, it's called. These are the most common primary tumours in the central nervous system at the Alfred Hospital, because we're an adult hospital, needless to say. And it would be not uncommon. It would be the same any major public hospital, if it's an adult hospital, to have this kind of tumour group as the most common primary central nervous system tumour. And they can be broadly categorised into 3 groups now. And this is a big change to where they were before. So one is a tumour derived from the cell called an astrocyte. It's called an astrocytoma. And these tumours have to be, have to be so-called IDH1 mutant. So that's one category. The next category is a tumour called an oligodendroglioma, and these are also IDH mutant, and they have 1p19q co-deletion. And finally, there's a glioblastoma, which is IDH wild type. It sounds pretty basic and easy. I read this and I think, what's the problem? Why do I have so much difficulty? Because it might sound easy when you see this print, but I can assure you, when you're trying to deal with tiny fragments of tissue, or tissue that's not quite in the tumour, or tissue that's purely necrotic, it's not quite the same ease of just reading these 3 categories. But the point I'm trying to make is that with time and with this 21 category, we're now into these three main tumour types, and that is not what it used to be. So it's a very interesting evolution to me to see how it's starting to tighten up over time in terms of what we can tell the clinician and how accurate we can be. And it's, yeah, there's lots of interesting aspects of this. So let's just have a look at some of these, because I have to introduce you to my wonderful world of pathology. And here is a classic astrocytoma IDH mutated. So these tumours occur, as I said, in adulthood, and they have a characteristic radiological appearance, and Mark and other clinicians can tell how they usually present. But what happens is that usually have a diffusely infiltrating pattern in the brain. Now, as I said prior, obviously, if something's diffusely infiltrating the brain, it's not very easy to go in and cut it all out. And it very much depends on where the tumour is to how easy it is to resect. But that's another issue. Let's have a look at the tumour. As you can see down in the pink slide here, the H&E shows that it's a very cellular tissue, and there are these cells all scattered through it. Now, these are the neoplastic astrocytic cells. And you can see down below, there's an immunoperoxidase for IDH1, and it's showing strong positivity. So the other point is that the ATRX here should be positive in the nuclei of these glial cells if it's normal. When it's lost, what it means is the IDH is usually being mutant. So these are kind of correspondingly negative and correspondingly positive. So this was, you can see, was a 31-year-old male. That's about the age group that these people get. This is about the age group that the people get an astrocytoma IDH1 mutated. It's a bit broad spectrum, but it tends to be those in that kind of age group. And the tumour has a slowly progressive clinical pattern over time. It's not rapidly progressive. It's usually slow progression over 3, 5, 6 kind of years. So for the diagnosis, you can see I've got my morphology, I've got my immunoperoxidase, and I'm happy to reach my diagnosis on the grounds of this. So I'm getting all this additional information, and I can get there pretty fast in terms of reaching a diagnosis, assuming everything seems to go according to plan. So there's my first category, astrocytoma IDH1 mutated. Now, the next thing about these is that when they are astrocytoma IDH mutant, I need to give them a grade. I told you previously how the grading system works in terms of a grade 1, 2, 3, 4, and a grade 4 tumour is a much more aggressive type of tumour. So we've always subdivided our astrocytomas into WHO grade, and it is important to kind of give an indication to the clinician how that patient is likely to progress, what kind of speed it is going to be progressing at. So you can see here, current WHO says if it's a grade 2 tumour, I use morphology, and I make sure that there's no CDK N2A mutations present. So I have to do something to say that the CDK N2A B or B mutations are not absent. So we do an ISH screen, and as you can see here on the right hand side, that's an in situ hybridization, and if you can see the green dots, that's controls, and the red dots are the actual CDKN2A. And so you can see that it's evenly placed, 2 and 2. And so people keep interrupting me. You can see that they are evenly spaced, and there is no evidence of any CDKN2A loss. So in that category, I use morphology. I say that there's no loss or mutations present in CDKN2A, and I can reach a diagnosis of grade 2 or grade 3 tumours. However, if I detect a loss, and if I do NGS and also pick up that there are mutations, then this tumour will behave in a much more aggressive fashion, in a grade 4 fashion. So it is important to stratify my diagnosis and my prognostic criteria based on what I'm seeing. So that's just an indication of how I would run through an astrocytoma. So this next tumour, oligodendroglioma, is also mutant when I do the IDH immunoperoxidase, and I find that 1p19q is co-deleted. So I look at the morphology, I look at the immunoperoxidase pattern, and classically they are IDH mutated. They have ATRX and p53 is lost. So I know the kind of pattern I'll be expecting. I do in situ hybridization for 1p19q, and I find there's co-deletion. And I can do next generation sequencing as well, because sometimes the IDH1 is negative, but everything else points to this being an oligodendroglioma. I will just tell you about that in a few slides' time. So let's look first at the beautiful pictures that I see when I find 1p19q co-deletion. And you can see here's 1p, 19q, and you can see that there's deletion. Well, a lot of these cells are deleted. It doesn't have two sequences of the red dot here. There's deletion of 1p, and here's deletion of 19q. And when I get deletion across both, it's co-deletion, and I know that I'm dealing with an oligodendroglioma, or at least it will support my other findings. So I did tell you that on occasion, my IDH1 is negative on immunoperoxidase. And that's interesting, and part of this talk will look into that a little bit further. But the thing about that is that I can only look at IDH1 using an antibody binding in immunoperoxidase. I don't look at IDH2, and IDH2 is present, mutations are present in about 10% of oligodendrogliomas. So that's when I'll do next generation sequencing, because we've got that on our panel, and you can see there's somebody who's turned out with an IDH2 mutation, which would account why the IDH1 is negative. So I just have to do those formalities to reach the diagnosis and make sure that I'm correct and everything's working for me. And it's been a delight for me to be able to get to this stage and actually verify that what I'm seeing is an oligo. The IDH1 is negative only because it's actually IDH2. So I've enjoyed this process and this learning. And often I've had these thoughts prior as to why this is not working, and now I've got the proof with the NGS as to why it's not. And I'm going to show you some more detail about that later. So let's come to this last tumour type. I really don't want to bore you with all my pathology, even though I could talk about pathology all day, every day. However, I do want you just to have a vague idea of what I'm talking about. And this is the glioblastoma. Now, for every research scientist out there who's listening, this would be the tumour. If we could find a cure, it would be just extraordinary. It's just this incredibly aggressive tumour. It occurs in adults. This person is particularly young at 35. It's usually more commonly a tumour of people in their 50s and 60s, but it is an aggressive tumour, and the patients can die within a fortnight of this diagnosis. And it just explodes in the brain and infiltrates throughout the brain, and it's incredibly difficult and incredibly sad. I don't really know how the actual clinicians who are looking after these patients deal with it. I just have to look down the microscope. However, this is the kind of tumour, and you saw previously the two other tumours just on H&E, and even if you're not pathologist, you can see that this cellularity is much, much worse. There's a lot more cells. There's a whole lot more variation in the cell size. There's a little bit of necrosis here, which is very classic. And these cells here are prominent because their vessels are proliferating under VEGF stimulation. Now, these tumours, when they can be glioblast, they are usually IDH wild type, and you can see that that is negative, and I've been showing you positive IDH previously, and here you can see the HRX is preserved. So this is the kind of classic pattern for glioblastoma, classic morphology, classic IDH, classic ADRX. But we are also doing next generation sequencing on all those who are under 55, because for a lot of reasons, we want to understand this diagnosis better, and we want to make sure that we're looking for all the mutations that are present and trying to understand. I think I'm just repeating myself, glioblastoma is better. I've wanted to do that kind of all my existence. And they do have a classic gain of chromosome 7, loss of 10. They have TERT mutations. They have often got RB1 mutations and NF1 mutations and EGFR mutations. There's a very classic kind of pattern of what they can have. They can often have multiple mutations. That's very, very common as well. So in the glioblastoma, I'm not doing it so much for diagnosis, like I've told you about in the others, but I am doing it to learn more about it and to see if there's any mutations here that are worth targeting for the clinicians. Or the more we do, the more we learn. So it's a space that's evolving. Now, one of the good, relatively good areas in this is that there is a clinical trial around within which if you look for EGFR and you find that it's amplified, potentially these people can go on a trial. So this is an example of when I do EGFR for these glioblastomas, and you can see that there's massive number of EGFR red dots, which indicates this patient is amplified for EGFR and may be suitable for this clinical trial. So in this circumstance, it's not for diagnostic reasons, but in this circumstance, I'm doing these additional tests to see if there's a valid clinical trial. So it's a bit varied. Another important test that's come online, and it's not done by us here, we send it away because I haven't got it, time is of the essence, but MGMT methylation is a useful bit of information for the clinicians, and there are various methods, but in essence, we're not running it here at the moment, and we just send it away to be done elsewhere. So I've mentioned to you some of the reasons why next generation sequencing is of use in terms of my diagnosis or in terms of offering clinical information, but there are other reasons why I have done next generation sequencing over the last 2 years or so since we got it up and running. And one of them is, and it equates with what the WHO says to do, is confirm a mutation that's very common in that tumour. So some mutations, some brain tumours, and for want of what it's called, a pleomorphic xanthoastrocytoma, commonly have a BRAF mutation. So I was expecting it to be there. It helped me with my diagnosis, and that was great. It was a BRAF mutation. But on the other hand, and you can't say just because it's a BRAF mutation, it doesn't mean it's that diagnosis. Because BRAF mutations, as you all know, are very common in many other tumour types, particularly in melanoma. So it helps me, but I have to see it in the clinical context, the full pathological context, but it helps me. It's a little bit of additional data to help me reach my diagnosis. So one is to confirm a common mutation in a tumour, and these are often rare tumours. Another one is to detect a specific mutation, which will allow me to meet a specific CNS criteria that is actually dependent on that mutation. So there's a type of tumour called a diffuse hemispheric glioma, this H3G34 mutant. And there's nothing I can do down the microscope other than to say it's a glioma to reach this specific diagnosis. The only way it is to do NGS analysis and detect it that way. So this is completely new in 2021, this diagnosis. But it's quite tricky because it just looks like a glioma. So you have to order the right test at the right time. So there's an example of when it's just a specific mutation, which will allow you to reach a specific diagnosis. And also here, you can do it the opposite way around, is to use your NGS to exclude a specific diagnosis. So in the pediatric world, there is a type of pediatric high-grade glioma. Obviously, we don't see the pediatric tumours, except sometimes they extend into teenagehood, and we do see people in their late teens. And in this category, they have to be wild type for H3 and wild type for IDH. So here are some more important reasons why we're using NGS for diagnosis now, to reach diagnosis, help diagnoses, and exclude diagnoses, all of which are new since 2021. So I decided, for this purpose of this talk, to put in a little action plan I've had in my head for a while and look at all the reasons I've done an NGS since we started NGS at the Alfred Hospital in December 21 for gliomas. This is just for gliomas. I wanted to see why I had done it and had it made a difference, and overall what was happening. So I selected the cases, and in general, as I've said previously, most of the reasons I do NGS in this context would be for diagnostic reasons, sometimes for prognostic, and I've shown you some of them, and sometimes for potential treatment. I wanted to have a look and just see exactly what had happened over the last period of time. So 42 cases have had NGS, which were called gliomas, and that's about 50% of all our glioma cases during that time frame. And it's important to realise I don't always do it, because there are circumstances where I absolutely don't need to. I've got the diagnosis already, or it's something like a glioblastoma, that tumour which is very aggressive in someone who's over 60, and we don't routinely do it because it's not going to offer all that much information. So it's a selective, and that's important when you've got a test that's expensive, and there are other reasons to be wanting to run the NGS. because we use the NGS sequence in that 1 week for all our solid tumours as well, not just CNS. So we have to be selective. So I've been selective. I've selected out 50% of our tumours for NGS. Okay, so I wanted to then break down why I had looked at these NGS, why I had done NGS, why had I performed it. And in the end, I've broken it down into 2 broad categories. And I'll show you some of the data, but it may not be all that helpful. But if in effect, what I did in 80% of the cases, I effectively said to myself that by analysis of what I'd done, what I would have diagnosed before and without NGS, I've come up with that it has helped my diagnosis in 80% of cases. It's either helped me to reach my diagnosis, which I thought would be there, or it's upgraded or provided diagnostic information. So that's a pretty high category of 80% and makes the test to me very useful. And this other issue, which I wanted to know exactly how valuable it was, and it's when I've already selected out cases that need NGS, you know, so this is not, as I said, not across all our gliomas. So I've selected out cases. This for one reason or another why I think we need to do NGS. And here is an interesting statistic that I found, that there was some variation in my diagnosis once I did the NGS. If I hadn't had NGS, I may have reached a different diagnosis, or I couldn't have reached the final diagnosis. So I think that's these 2 points are very, very valid and show how relevant and how integrated the molecular testing and using NGS has become within this at the Alfred Hospital. And now this is a bit more complicated, and I think it's probably more applicable just to people who deal with this all the time. But I've just, instead of just giving you those overall statistics of how it worked in terms of allowing me to reach a diagnosis, or how it confirmed what I'd already thought, I've just given a breakdown of all the different NGS uses that I had. For instance, there were some examples where the immunoperoxidase with IDH on something that I thought looked like a standard astrocytoma and expected to be mutant should have been mutant. And so I confirmed it was actually mutant. In fact, for some reason, the IDH immunoprotection hadn't worked. So that was great. It confirmed what I thought, and without it, I couldn't have reached the CNS criteria currently. Here's another one where these are all the ones that I've highlighted, the ones where I actually got new information that changed my diagnosis somewhat, but all the ones that are not bolded are all the ones where I actually got what I really expected. So I got an IDH negative case, but it was mutant oligodendroglioma in 3. And I illustrated how that could have been an IDH2 mutation, so that's to be expected. The GBMs, of which there was the bulk of the tumours that were done, were GBMs, because GBMs, glioblastomas, are the most common tumours. No change to the diagnosis was received, so it remained status quo, and it was a GBM. And then I had a couple of cases where glioblastomas were in a very unusual site or had very unusual morphology, and the NGS helped me to confirm that I was right, even though they were a very unusual site for a GBM, or their morphology was not usual. I was correct in terms of what to expect in terms of a GBM type of molecular patterning. And in some circumstances, the additional mutation information helped me support my diagnosis. So they're all very fascinating and interesting and relevant. But I think the thing that really, the one that, and I'm still finding this interesting, and I really want to follow these cases through, were cases where the tissue I received didn't match what the imaging was. And this is an example of where the forum to discuss radiology, clinical and pathology is so important, because I had little biopsies, and for those who don't realise, they sometimes just take stereotactic brain biopsies, which is just tiny cores of brain tissues. And I had little biopsies, and in the little biopsies, I could barely reach a diagnosis of an astrocytoma, and yet the imaging showed this massively widespread tumour. And on these samples, I did NGS, and on the NGS, I found that their molecular profile, despite what I was looking at down the microscope, was that of a glioblastoma. And that kind of patterning matched the imaging patterning. And I'm really, really still, find this a very interesting area. And there's a little bit of literature coming out on it, not much. There are other people who are seeing similar things, and I meant to chat with my colleagues overseas at the time. But the principle being, what is interesting here is what we're seeing is that the NGS, the next generation sequencing, is really providing us with, in the right circumstances, valid information and very useful both for diagnoses and prognoses. And it has to be selected for carefully, and you have to interpret that data carefully. It is just more data. And as my colleagues overseas say, that as pathologists, what we are now are becoming kind of data repositories. We know how to interpret data. We know the tests that are being performed have been NATA accredited. We're in charge of them. We know that they're valid. And we put all that data together. And with that data, we can provide a much more accurate information to the clinicians. So I just wanted to put this in. Lucy Gately, one of the medical oncologists, and I have been looking at this, and also Hayley has been doing it in terms of looking at some of the clinical data. And we're still going to we're going to hopefully put some of this together and make some sort of overall comment about treatments and the use of NGS. But I just wanted to show you, because at the very beginning I said that there were three main reasons we did NGS in central nervous tumours. One was for diagnostic, and I've proven to you how valuable that's been. One was prognostic, and I've shown you some examples of why prognosis is important. But here's another arm, which was the clinical. Now, I asked Lucy about this, and I'm afraid I've mentioned to you already something about the role of amplification of EGFR, and whether all this information I'd given on, for instance, the glioblastomas had actually led to any considerable change in the treatment arms. And I don't think there've been that many examples out of the role so far in which there's really been a major change, but it may be that trials open up. And it may be that there was a case that I haven't gone into in any detail with you guys, but it may mean that there's some cases that immunotherapy may be useful in the circumstances if they recur. So I don't want to go into that aspect, because I don't talk about things that I'm not au fait with, and this is the clinical role. But overall, despite the potential for NGS to open up treatment arms, the area of brain tumours is still evolving, and there are not that many treatment arms. Let's hope that the treatment arms open up with time, but currently it's not as clear-cut as the use of treatment for melanoma with a BRAF mutation, for example. So this is still up and coming. There are still potential trials happening, and I hope it will evolve. So I'd finally like to say that all this information and immunoperoxidase and in situ hybridization is done by Andy Diamond with an N at the end. I'm very grateful for him, and we've established that testing structure, and we bought a specific machine that so we could do our immunoperoxidase in situ hybridization and load them at the same time, and it can come in it can come out at any time of the day, which has been fantastic. I also want to thank everybody who's in the molecular pathology team at the moment, Adam and Nick and Rebecca and James and Alicia and Rod, all of whom work very hard to interpret all the VAFs. And I'd like to thank all my colleagues in the medical oncology team who primarily work within the central nervous system, and who've been, we've been talking with, as I said, Hayley Roberts and Lucy and I've been talking about this, and also Mal Amaratunga, who is always in these discussions at the clinical meetings, and is very much involved in looking at the role of new molecular mutations in NGS, in central nervous system tumours. So with that, I would like to thank everybody again and see if there are any questions out there. And I hope that it hasn't been too confusing entering all these complex medical terms into a global discussion.
Thanks so much, Catriona. Fantastic overview of the sort of status quo of modern management of brain cancers, which is increasingly dependent on not only pathology, but of course, molecular pathology. Are there any hopefully we've got a few brain cancer interested researchers dialed in today. Are there any of the dialed in have a question for Catriona? just put your hand up or write something in the chat.
There's one chat something.
Oh, that's just an invitation to ask question.
Okay.
Catriona, what do you? I mean, you're obviously a keen and long-term observer of this disease.
Mm-hmm.
What do you think are the, and obviously the genomic subclassification of brain cancers generally has I think helped to clarify it's obviously the subtypes and the clarity of classification, I suppose, has evolved fairly well.
Mm.
What do you think is the next, like, how do we kind of harness this, how do we harness this information? I mean, I'm just thinking back to melanoma. Yeah. So we, there was that landmark paper in about 2001 or 2002 that really was the first, targeted sequencing paper that discovered the BRAF story. And that obviously led to the development of BRAF inhibitors, particularly around that. What are the targets in melanoma, brain cancer? And brain cancer is such a heterogeneous group of diseases.
Yeah, I think that's the issue, isn't it? I think
lump them all together. Where do you think the really promising targets are from a therapy development point of view, particularly for a basic research audience like we've dominantly got here?
Well, I think that's a pretty tricky question, Mark. I think the most difficult area, without a doubt, is the glioblastoma. I think that they have such a spectrum of morphologies, which means they've got such a spectrum of mutations that they are going to remain our biggest problem into the future. And although it is the most common tumour as primary brain tumour, so to me, trying to work out novel ways to approach glioblastomas would be the most important thing for researchers to going forward, because it is so common. But do I know where that aim should be aimed at in terms of what the NGS? And I think the NGS is opening up more questions about the actual broad spectrum. I think we can only push on and just see because some of the glioblastomas are starting to categorize into groups. So rather than saying, oh gosh, it's this huge big field, what we're starting to see is that there are glioblastomas, and there's 4 subtypes of glioblastoma. So the question is if you can find the drivers in perhaps those 4 subtypes, and this whole molecular profiling is allowing us to look and see whether, in fact, although it's possibly not this huge amalgam of different types of tumours, stem cell driven, but perhaps we've got a bit of leeway there in terms of subtyping of the different tumours, and maybe we can use NGS to be more specific in terms of glioblastoma. There's also the use of specific mutations in when you've got, I think Mal and Rotunga's been looking into this a bit, whether you can target those things like PXAs with a BRAF mutation when the BRAF mutation is there. So there's some evidence for that, but obviously the clinical trials aspect is more in your framework than mine, I think. But I do know a lot about glioblastomas, I think, over time, and if there are people who are there, I'm more than happy and very willing to sit down and discuss with researchers some of the information that I've gained over the last umpteen years in terms of...
Jack Edwards, you got a question, Jack?
Yeah, thanks, Mark. And thanks for the talk, Catriona. I was just curious to get your thoughts on, there's been a bit of noise around whole genome methylation arrays for characterizing brain tumors.
There has. There's plenty of noise, yeah.
Yeah. Do you think that's a realistic and maybe valuable addition to your toolkit? Do you see that becoming part of
the CNS methylation is only currently a desirable category in terms of the how we're going. So it will become more prevalent, I think, and it seems to be pretty accurate and good, but it's very expensive, it takes time, and we've got to just balance out what can be done locally and how much additional information can be provided. So we can do the methylation profiling, we actually can do it, we can add it to the test, but if you can do cheaper tests that are more relevant to you and get the same information, I don't know what the relevance is. So a lot of my colleagues would say, well, you know, you'd only use that testing structure if you really were at a loss as to where you were in terms of diagnosis. So to me, and it might evolve differently over time, but I went and spent my sabbatical over at the John Radcliffe in Oxford, and they were doing it along those lines, the use of this methylation profiling in those circumstances where things aren't kind of marrying up. So at the moment, I think, apart from if you live in Germany and you're at the Heidelberg Centre where it's evolved, it's not freely available, it's very expensive, and a lot of diagnoses can be reached without a need for it. So to me, let's call it a fabulous new test that has benefits in certain circumstances and could be very useful, but is not freely available.
Thanks.
There's a question here from Sal. Why do you think the pediatric gliomas have such different mutational profiles, I assume, compared to adult? And is this reflected in their morphology, i.e. are adult and pediatric gliomas distinct under the microscope?
A lot of pediatric tumours have very, very different mutational profiles to adults. I think there's a completely different driving circumstance in these pediatric tumours. Are they different morphologically? Not so much. A little bit. I'd say subtle morphologic differences and subtle mutational profiles. Can I always distinguish them? No. Some of the gliomas that but as I said at the very beginning, gliomas tend to categorize into where they're found and at what age they're found. So there are specific tumours in the pediatric age group that only ever occur when people are under 1 year old, and they only ever cited in site X. So you can't take individual bits of information. You have to take all the information you have available when you're looking at any type of tumour. So are they distinct? Well, I don't just look under the microscope. I find out what their site is. I look at the immunoproliferation profile. In some circumstances, though, morphologically, there's a considerable crossover between some of the pediatric tumor gliomas and adult gliomas. That's all I can say. I think it's probably to do with their basic mutational profile, where it's coming from. And like all pediatric tumours, there's a whole lot more mutational profile in differences to do with, I think, stem cells in the young age group. It's, to be honest, pediatric gliomas, there's not a lot. I don't see a lot of pediatric gliomas, so it's not my specialty.
Okay. I sort of noticed in a lot of your slides you had reference, when you speak of reference to NGS, you were focused probably a bit more on point mutations, identification of that as a tool to identify point mutations, whereas copy number change you tend to use.
Yeah, it's not, your copy number change is not really in the kind of diagnostic profiling that we need to do to make a difference. So it's.
So do you get in your standard workup, do you derive copy number information from?
We can with our machine, but we haven't got it up and running. It's just one of those things, I have a limited capacity in terms of scientists. They're working full bore doing all the mutational profiling for the hospital and also for the Northern, so that there's no time to add that CNVs. And if it's not going to be of direct clinical use, I'm not pushing it. So it's available somewhere, but we haven't got it down to the level of being able to present it as accurate NATA accredited clinical data. Yeah,
that's fair enough. Well, that's an interesting comment though, because sometimes the pathology drives the clinical use. It doesn't always go in the other direction. So because the pathology identifies the change that gives you the clue that you need to actually develop a therapeutic approach that's relevant to that particular change. So great. Okay, any other questions at all? All right, well, we're pretty much out of time, so it just remains to thank Catriona so much for her presentation and updating us all on the kind of status quo of the role of molecular pathology, particularly in the diagnosis of brain cancers, and also obviously to thank everyone for attending today and for your comments. This is, and also to advertise the next seminar, which is actually this week. We must be having a bit of a rush of these before Christmas, which is great. This will actually be on Thursday this week. I presume it's at the same time, is it, Hannah, around midday? And the guest presenter will be Mitch Lawrence, who will, I would argue, somewhat controversially discuss the potential use of testosterone to treat prostate cancer. Will surprise many of us who spend most of our lives trying to suppress testosterone in the treatment of prostate cancer. So we really look forward to your talk, Mitch. And thanks everyone again for dialing in, and have a great day.
Thanks, everyone.
Thanks very much.
Bye.
Thank you. Bye.